Bom dia a todos. Esse é o 13º ano deste curso. Estou muito orgulhoso de fazer parte deste curso cada ano. Obrigado ao meu querido e lindo namorado, Dr. Esper, por fazer tudo possível. Ok, hoje irei falar sobre os obstáculos para a cura do HIV. So we have an amazing armamentarium of agents that can be used to inhibit HIV. And that has really transformed the outlook for individuals living with HIV/AIDS. In fact, I heard an amazing statistic from Chip Gulick in New York. And if you are HIV positive and you're in care, meaning that you make frequent visits to your physician. Standard of care in the United States is 4 visits per year. Your life expectancy is longer than someone who is not infected, but who is not in care. So the take-home message is that the medical profession works. Making regular visits to your doctor actually does extend longevity, because in those visits, your doctor is more likely to catch early detection of those diseases that ultimately lead to morbidity and mortality, such as cardiovascular disease and type 2 diabetes. So if we have these amazing drugs, and they can essentially extend longevity almost as good as someone who's not infected, then why are we not happy with this situation? Why don't we just stop there? And the reason is that in individuals who are on antiretroviral therapy, there is immune inflammation. So the immune system is continually sensing the presence of something, even in individuals who are on effective antiretroviral therapy. And people who have participated in this sort of research include Tim Hunt at the top and Steve Deeks, who I understand is here for the course this year and participated in a satellite conference organized by the American Foundation for AIDS Research, AMFAR. So hopefully some of you made that conference. And what Steve Deeks has shown is that if you look at untreated individuals, as measured by the frequency of HLA-DR CD38 positive CD8 cells, there's a lot of these cells present in untreated individuals. And the frequency of those cells, the frequency of immune inflammation, is reduced in individuals who are on antiretroviral therapy, but it's not normalized to levels seen in uninfected individuals on the right. And it's that immune inflammation environment that creates the comorbidities that limit life expectancy in HIV infection. And what's essentially happening is that young people are developing diseases that are more common in older individuals. So HIV/AIDS is essentially an accelerated aging disease. So for this reason, the field has embarked on a bold journey, a bold quest to try and come up with a cure for HIV. Even though we have a very fortunate situation where there is very limited pill burden. 1 pill a day is the standard of care now. So even though we have this very limited pill burden, it should be easier to maintain antiretroviral therapy, there's still a high rate of treatment failure because of pill fatigue. For those of you who've had a bacterial infection and have to stay on a 10-day course of antibiotic, that's a very, even though it's just 10 days, most people will stop taking their pills after 7 days. So imagine having to take a pill every day for the rest of your life. It's not an end solution. And that's why the object of the cure is to get to zero medication, medication-free remission, or eradication of the virus. Now, unfortunately, HIV has come up with a strategy to persist in individuals who are on antiretroviral therapy. And irrespective of the time in which you've been on these antiretrovirals, if you stop taking the medication for whatever reason, invariably the virus rebounds very rapidly. Usually between 2 and 4 weeks, 90% of the individuals will have rebounded. So even though the virus is undetectable in individuals on antiretroviral therapy, it's still there. And the big drive in the field right now is to try to understand where is the virus hiding in individuals who are on therapy, because that's really key to coming up with those sorts of strategies that could be used to eliminate those reservoirs so that we can achieve a cure. Whether that cure be functional, the virus is still present, but the individual does not have to take the antiretroviral therapy, and the individual does not transmit any virus that's still present, or ultimately a sterilizing cure, where there is no trace of any viral nucleic acids in any of the tissues. And obviously that's a major challenge facing scientists today. So most of the attention in terms of how HIV persists in individuals on therapy has focused on the phenomenon of viral latency. Latency is a process that is well recognized amongst virologists. There are some viral families, the herpes viruses, for example, that have evolved proteins that force the virus to become latent when the environmental conditions are unfavorable for viral replication. But HIV doesn't have any of those elaborate mechanisms used by some of the larger viruses, such as the herpes viruses, because HIV is a retrovirus. It has far fewer genes. It's 9.7 kilobases, so it's far less genetic space to squeeze in the sorts of functions that might promote viral latency. So what individuals such as Doug Rich and Tony Fauci and Bob Salesiano here have shown is that within infected individuals, there are memory CD4 T cells, and those memory CD4 T cells contain the virus in a latent state. Now, this latent infected cell is really going to be a very difficult cell to eradicate. And the reason is that if one looks at an infected cell on the right, there's many things going on. Viral RNA is being produced, viral proteins are being produced, the virus particle is budding from the cell surface. It's easy for the immune system to recognize this cell as being different. However, this cell is really not different to a cell that is not infected. There aren't any viral transcripts, at least not according to conventional models of latency, but we'll discuss that a little bit more shortly. And there are no viral proteins that can be sensed by the immune system as marking the cell for elimination. So it's generally considered that this is the single biggest obstacle to the eradication of HIV. And some of the speakers at the conference will talk about some of the clinical studies being explored to try and remove this type of reservoir. But the early studies arguing that the majority of the latent reservoir was perhaps static, not changing over time, is being re-explored and re-investigated in work from people like Mary Kearney at the NCI, together with colleagues John Coffin, Frank Maldarelli, John Mellors and colleagues. And what they found is that the process of homeostatic proliferation that was first identified actually by Rafik Sekeli and Nicolas Chaumont when they proposed that infected cells may be replenished through the process of mitosis and homeostatic, as a normal part of homeostatic maintenance of cell number. And what Mary Kearney and colleagues have done is taken this one step further to show that some of these viral genomes from a latently infected cell that are duplicated are actually participating in reservoir maintenance. Because when this phenomenon was first described, most people thought, well, perhaps this process favors the duplication of viruses that have many defective defects in their genome. They're incapable of making infectious virus. So what's being duplicated is a population of non-functional viral genomes. But what Mary Kearney has shown is that not only do functional viruses get duplicated by homeostatic proliferation, but they actually get expanded over and over until they represent a major part of the overall virus population. And the reason is that some of these viruses, when they integrate in a cell, they integrate next to a gene that is involved in cell cycle control. Insertional activation, a process that's been studied extensively in cancer-causing viruses such as murine leukemia virus. Here you have a virus with a very powerful promoter, enhancer, an LTR. It integrates close to a gene that's normally involved in regulating cell cycle. So now the normal process of cell cycle regulation is taken over by the virus, and the virus drives proliferation of the cell. So if this original parental cell contains a genome that's integrated next to a growth factor gene, then that cell is going to have a growth advantage over other infected cells, and it's going to be expanded over and over again. In some individuals, these clonal expansions of proviruses can represent 30% of the overall virus population. So with this incredible amount of insertional activation going on in HIV infection, the question has always been, well, why don't we see more cancers? Why don't we see more leukemias in HIV-1 infected individuals because of this potential for HIV to integrate near a growth factor gene or a gene involved in cell cycle regulation. So at any point in time, 5% of these expanded proviruses are also transcriptionally active. And this has also raised some awareness that perhaps this reservoir is not as static as originally believed. And so those active proviruses, one would expect, if they become capable of making viral proteins, then the immune system would clear them. But as recent work presented at CROI demonstrated, some of these proviruses can go through cycles of expression. It's called waxing and waning. They get turned on to express the virus, and then they return to a resting state. So one of the take-home messages from this type of research is that perhaps even the latent reservoir does not remain in a latent state forever. That there are episodes with which cells within the reservoir become stimulated perhaps through cytokines in the environment. They express the virus, they don't get killed, by immune clearance, and they return to a state of latency. And obviously that provides an opportunity for investigators to find ways to attack the reservoir whenever reservoir cells become visible to the immune system through these episodes of expression. One of the less popular mechanisms that's being explored to explain how HIV persists is this phenomenon of ongoing viral replication. Again, at CROI this year in Boston, there was a lot of debate over these different mechanisms for viral maintenance. And this particular mechanism is, I think, misrepresented by the term ongoing viral replication. I think this is not a proper way to describe this sort of mechanism, because for virologists, ongoing replication means that a cell becomes infected on the left, it makes virus that goes on to infect another cell, and this process is repeated successively over many cycles. But there is no evidence for this sort of successive spread of virus in individuals on antiretroviral therapy. However, there is good evidence that this occurs, single cycles of infection, where a cell is making virus, even in individuals on therapy, and some of that virus is able to make it to another cell and reverse transcribe. And in virologic terms, we call that single cycle infection. The difference is that single cycle infection on the left would not be expected to lead to a significant degree of virus evolution or the creation of mutations that lead to drug resistance. And that's one of the big arguments against an active viral reservoir in individuals on therapy, that people say, well, if the virus is replicating in the presence of antiretroviral therapy, we should see drug resistance. The other thing that this mechanism has to contend with is the fact that there is the presence of these amazingly potent antiretrovirals. So how would the virus be able to do this in individuals who are on effective therapy? And Ashley Haas and Tim Shacker and Courtney Fletcher have been exploring how the antivirals penetrate the different tissues within the body, because what they hypothesize is that there are some tissues, perhaps the lymphoid tissue in gut-associated lymphoid tissue, that may not be as accessible to antivirals as we see in the blood. And what they have shown is that the concentrations of lymph node in terms of the ratio relative to blood is much lower than the concentration in other tissues or even in peripheral blood. And they've argued for the presence of pharmacological sanctuaries where the drugs are not accessing all parts of the body equally efficiently. Now, there's a good slide to illustrate this phenomenon. For those of you who enjoy drinking beer, there's a few in this audience, I'm sure, they may remember Heineken. Heineken was very popular in Scotland when I was a student. In fact, my university, a pint of Heineken was 5 cents. It was subsidized. Usually, if you go to the pub next door, you would pay $1, right? So there was an incredible incentive for the students to stay in the university and drink in the university premises. It's actually amazing that anyone managed to matriculate when the beer was subsidized like this. So anyway, here is Spock from Star Trek, who's feeling very depressed. His girlfriend has just left him, taken the dog and the cat, and his ears are drooping. And then he takes some Heineken, and Heineken refreshes his ears, and now everything is as it should be. So the moral of the story is that Heineken refresca as parte que outras cervejas não conseguem alcançar. So the moral of the story is that, according to Tim Shacker and Ashley Haas and others, that perhaps some antiretrovirals do not behave like Heineken, where others do behave like Heineken. They reach parts of the body where the virus is hiding. And so exploring the different classes of antiretroviral agents for those that may be able to more effectively penetrate lymphoid tissue is an exercise ongoing in a number of laboratories. Now, in addition to these reservoirs, these long-lived reservoirs that are maintained by latency, by homeostatic proliferation, perhaps by de novo infection, new infection of cells, we also have a problem measuring the virus in individuals who are on antiretroviral therapy. So if you're going to start clinical trials to examine how effectively you are eliminating the reservoirs, then you should have some very sensitive ways to measure the reservoir so that you know you're doing a good job, so that you know that your treatment is actually reducing the reservoir. And that is where the field has really suffered a major sort of setback, because most of the approaches being used to measure reservoir have not really been validated. For example, the gold standard is viral outgrowth assay, where you take cells from an individual, you do limiting dilution, and then you rescue the virus from those cells after ex vivo activation. But those are incredibly laborious and they lack precision. And if you have clinical samples where cell number is limited, you can't do this type of viral outgrowth assay. So people have been looking for surrogates, ways to measure the reservoir that could be informative and could be biologically relevant. One of the assays, one of the surrogates that's actually gaining some favor is cell-associated viral RNA. Now, cell-associated viral RNA is not a new phenomenon. Steve Walensky and colleagues in 2009 demonstrated that if you take individuals who are on effective therapy and look for the presence of the viral RNA in their cells, it's readily abundant, it's readily detectable. But no one really paid much attention to this or as to whether it might be biologically significant. So Jonathan Lee and colleagues have tried to assess whether this cell-associated RNA has any biological significance. And what they did is they took individuals who were on effective therapy and who stopped therapy, and then they measured how long after they stopped therapy would the virus take to rebound. And then they compared the time to rebound with the amount of cell-associated viral RNA and proviral DNA in those individuals. And what they found is that the individuals who rebounded the fastest, the ones who rebounded in less than 4 weeks, also had the highest levels of cell-associated RNA. So they could predict how quickly someone would rebound by measuring their cell-associated RNA at baseline. And now a number of groups are trying to extend these observations to get more, to understand better what this might mean. Does this cell-associated RNA occur in latently infected cells, or is there different types of the reservoir? There are some cells that are always latent and don't make any RNA. Are there some cells that are in the process of continually transcribing? And that opens up all sorts of questions in terms of what the dynamics of the reservoir might be. So the size of the active reservoir is associated with the timing of viral rebound, and significantly lowers of cell-associated RNA are also found in individuals who start treatment very early, where presumably you're limiting the reservoir size. So this suggests that infected cells, if they're making viral protein from this RNA, might be recognizable by the immune system. And then, of course, the long-term persistence of HIV, in addition to being visualized in long-term clinical follow-up studies, has also raised its ugly head in terms of attempts to eliminate the viral reservoir. So Dan Kuritskas and colleagues, and Dan is participating in the conference this year, were trying to emulate what happened with the Berlin patient Timothy Brown. I mentioned him last year, an individual who underwent 2 bone marrow transplants to treat his leukemia, and this individual was also infected with HIV, so his physician, Gerhard Hutter, transplanted Timothy Brown with stem cells from an individual who had a CCR5 mutation, and the story is that Timothy Brown is now the only individual who has been cleared of HIV, to be cured of HIV. The man who once had HIV. And what Dan had wanted to understand is whether something about the transplant procedure itself might have contributed. In other words, what was the most important element? Perhaps the need for CCR5 deleted cells could be obviated if some of the cure was created by the process of bone marrow transplantation. So 2 individuals were transplanted with cells from individuals who had normal CCR5 alleles. And this is patient B. After the transplant, they examined different tissues for the presence of the virus, and to the limits of detection, which is 1 copy per million cells, they could not find any viral nucleic acids in this individual after the transplant. So based on that result, the individual who had been maintained on antiretroviral therapy was discontinued therapy, and everything looked great for the best part of almost 10 months. And around 210 days, the virus rebounded and was put back on antiretroviral therapy. So this suggests that the presence of CCR5 negative stem cells that were used to cure the Berlin patient seems to be a very important component of the cure, at least in the Berlin patient. It's still unclear where in these 2 individuals the virus rebounded, as the host CD4 T cells, which presumably would contain all the reservoir, were removed by the process of bone marrow transplantation and completely replaced with donor cells. So that brings me on to this topic of reservoirs other than CD4 T cells that might perpetuate HIV in the setting of suppressive antiretroviral therapy. And the question my group and many other groups have been asking is, are there other reservoirs other than memory CD4 T cells that might be involved in maintaining HIV in the presence of antiretroviral therapy? Now, HIV is a lentivirus, and lentiviruses in general have long been known to associate with macrophages, both in vitro and in vivo, especially in the non-human primate model. So most of the attention in terms of reservoir maintenance has been on the memory CD4 T cell reservoir. But we have to look back in the literature to realize that there are many characteristics of the primate lentiviruses that suggest that they have evolutionarily responded to the challenges of infecting macrophages. And there are a number of challenges to infecting macrophages, but lentiviruses appear to have surmounted those obstacles, meaning that they seem very determined to acquire the ability to infect this type of cell. Now, one of the obstacles that viruses like HIV will encounter when they infect a macrophage is a DNTP trihydrodephosphorylase called SAMHD1. And SAMHD1, its job is to reduce the concentration of deoxynucleotide triphosphates in cells that are non-mitotic. because in a cell that is non-mitotic and not synthesizing DNA, then those deoxynucleoside triphosphates are going to be excessive to need. They're not needed in the manufacture of DNA when there's no DNA being manufactured. So SAM-HD breaks down these DNTPs. However, viruses like HIV-1 and SIV require DNTPs to make cDNA. So normally, that macrophage with active SAM-HD is not permissive to infection by a virus like SIV. So what HIV-2 and SIV have done is they have evolved a protein called VPX. And VPX interacts with SAM-HD and drags it to the 26S proteasome for degradation. And once that happens, the DNTP concentrations increase, and then the virus can synthesize cDNA. Now, that's also been shown to be important in vivo. So people like Ron Desrosiers, who's participating in the course, has shown that if you take a virus like MAC239 and you delete the VPX protein, that virus now has a tough time infecting macrophages in the monkey. So if you look in the wild-type animal in the spleen, there are lots of infected macrophages and CD4 T cells in the spleen and the colon. But in the animals infected with the VPX mutant, you don't see infected macrophages in either location. So it's very important for viruses to get rid of this SAM-HD so that they can invade this cell type, which seems to be important for virus-host interaction. The conundrum, the paradox, is that HIV-1 doesn't have a VPX protein. So the question has always been, how does HIV-1 evade SAM-HD? How is it able to infect a macrophage if there is no DNTPs to allow it to synthesize its cDNA? In fact, some individuals have interpreted that as arguing that macrophages are not susceptible to HIV. Yes, HIV-2 and SIV can infect macrophages because they have a VPX protein, but HIV-1 is different, and it has not evolved the capacity to infect macrophages. And that's not true. So the question is, how does HIV infect macrophages in the face of SAM-HD restriction? Now, HIV-1 has a VPR protein. It's the ortholog to VPX, but we and others have shown that VPR doesn't have any SAM-HD eliminating capacity. So Baek Kim and colleagues at Emory University have hypothesized that the KD of HIV-1 reverse transcriptase is much higher for DNTP incorporation than HIV-2 or SIV. So that HIV-1, when it infects a macrophage, is able to use the DNTPs far more efficiently. So even though those DNTPs are at very low concentration, HIV can compensate for that. And again, that's something that we'll address shortly. So Tim Plitnick, a graduate student in my lab, has been trying to understand how HIV-1 is able to survive in this very hostile environment created by SAM-HD. And one of the first things that he showed is that if you take HIV and SIV infected cells and look at the concentration of SAM-HD, what you find is that in the HIV-1 infected cells here, there are lots of infected cells, and there's also lots of SAM-HD, this lower right quadrant. Whereas in the SIV infected cells, all the SAM-HD has been degraded by the process of SIV infection. So what this tells us is that HIV-1 is able to infect a macrophage, but does not change the level of SAM-HD expression. So he then asked, is it something about the reverse transcriptase of HIV, as proposed by Baek Kim and others, that allows it to use those DNTPs far more efficiently. So what he did is he took the reverse transcriptase genes between HIV-1 and SIV and exchanged them. He interchanged them. So now he created chimeric viruses. In this one here, this is an SIV backbone with the HIV-1 reverse transcriptase. So you'd predict that this virus should be able now to replicate in macrophages in the face of SAM-HD. This virus should not now need a VPX for survival. Now those viruses, those recombinant viruses, the reverse transcriptase gene is functional, as evidenced by their differential sensitivity to RT inhibitors. So SIV-RT is sensitive to tenofovir. It is not inhibited by nevirapine. And what he shows is that this recombinant virus has the same sensitivity to HIV. So in other words, this HIV-RT is faithfully reconstituted in this virus. However, that virus still requires VPX to invade macrophages. However, when he does the reverse experiment, what he found is that the SIV reverse transcriptase, when inserted into an HIV-1 backbone, now is able to infect macrophages even in the absence of VPX. So that suggests that there's something in the HIV-1 genome, something in the HIV-1 genome that confers intrinsic resistance to SAM-HD. But that still does not get at the main question, which is, are macrophages actually important in the maintenance of HIV persistence in individuals on therapy? It's a very controversial topic, but it's really made controversial because of the lack of information. It's very difficult to study macrophages because they are located in the tissues. So one of the approaches that we've taken is to try and understand the composition of the virus from the composition of, the composition of the reservoir from the composition of the virus that emerges from it. The capacity of HIV to infect a macrophage is dictated by how efficiently the viral envelope can use low levels of CD4. So we asked whether the existence of a macrophage reservoir can be inferred by the presence of viruses in plasma that are able to use the envelope, the CD4 molecule, very efficiently. And specifically, if an individual stops therapy, do some of those viruses that rebound have the characteristics of a virus that can infect macrophages? So here is the process of interaction with receptor and co-receptors. The virus first has to bind to CD4. That exposes epitopes on GP120 that then allow it to interact with co-receptor molecules such as CXCR4 or CCR5. Now, because macrophages have very, very little CD4, in fact, it's almost barely detectable by fluorescence-activated cell sorting. For a virus to infect a macrophage, it has to engage CD4, those rare CD4 molecules, very efficiently. So we hypothesized that when someone stops therapy, this virus that's rebounding may have that capacity. It may have that high CD4 affinity. So Viviane Machado, Carioca, a PhD student in my lab, has been spearheading this project to try and understand whether some of this recrudescing virus is comprised of macrophage tropic viruses. So she's developed a strategy where she takes the plasma from individuals and she clones the envelope after single genome amplification. Then she takes those individual envelope molecules and inserts them into an HIV backbone, rescues the virus, and then asks whether any of those viruses can replicate, can infuse with, and then replicate within macrophages. To ask whether the virus fuses, she uses an approach originally developed by Warner Green at the Gladstone. She modified it so that it will work on macrophages. So basically, you take a beta-lactamase enzyme and you fuse it with VPR. It's packaged in the virus particle. Then you introduce the substrate of beta-lactamase, which is CCF2, by simply adding it to the medium of the macrophages, and it's taken up by pinocytosis. If the virus fuses with the macrophage, the beta-lactamase is deposited in the cytoplasm, and then it acts on CCF2 to change its color from green to blue. So then you simply look at the cells under a microscope. If the cells are blue, it means that the virus had successfully fused with them. So she's done this on about 8 patients now. She's made about 2,000 recombinant full-length infectious viruses. And if she looks at the fusogenicity, what you see is that most of these viruses fuse with macrophages at various levels of efficiency. Now, if you take a virus that's clearly able to infect macrophages, and these are these ones here, these are recombinant, well-characterized viruses, ADA, 5002, YU2, they fuse very efficiently with macrophages. These are viruses that do not. These are viruses that will only infect T cells. They have low CD4 affinity. You will see that some of these individuals, the majority of viruses do not efficiently fuse with macrophages, but there's a subpopulation, about 1 to 3%, depending on the individual, that appear to fuse very well with macrophages, almost as efficiently as these control viruses. So she then focused on these ones at the top and asked, do they actually replicate in macrophages? Do they confer the ability to undergo spreading infection in primary macrophages? So here are the controls. This is the replication of these viruses in macrophages. The ones in black here at the top are those highly macrophage-tropic YU2 ADA 5002 viruses that I showed in the previous slide. The T-cell tropic viruses that are at the bottom here, they don't replicate in macrophages. If you then take these viruses derived from these patients, you see that they fuse and replicate very well in primary macrophages. So the conclusion is that macrophage-tropic viruses are present in individuals and they promote viral outgrowth when treatment is interrupted. So that infers, it does not prove that those viruses originated from macrophage, but at least it provides strong evidence that macrophage reservoirs do persist in individuals who are on antiretroviral therapy. And as a consequence, we're going to have to come up with perhaps completely different strategies with which to remove this reservoir, because the strategies being proposed and being pursued for the elimination of CD4 T cells are unlikely to work with this reservoir of macrophages. So I'll finish by acknowledging some of my collaborators in Barcelona, at Harvard, UC Davis, Mass General, in Florida, and particularly the efforts of Viviani here, Jen here, and Tim here. Thank you for your attention. I'll be happy to take any questions.
So Mario, maybe it's just the fact that I'm dim, but why is affinity for CD4 so central for infection of macrophages versus CD4 T cells? Yeah. There's plenty of CD4 on macrophages, sure, a lot less than on CD4s, but why is affinity not so important for infection of CD4 cells by those same viruses?
Right. So the vast majority of viruses in plasma in individuals who are not in therapy are T-tropic and have very low CD4 affinity because it's excessive to need. There's plenty of CD4 on a T cell, so the virus doesn't have to have that high CD4 affinity. Now, why CD4 affinity would be bad? Why wouldn't it just all have high CD4 affinity? is probably related to their fusogenicity. And the viruses that have a very high affinity for CD4 may just fuse so effectively with T cells that the fusion process is cytopathic. So we've noticed that those viruses that have that high CD4 affinity are extremely cytopathic for CD4 T cells. We originally thought it might be antibody sensitivity, that having a high degree of CD4 affinity might expose make the virus more vulnerable to neutralizing antibodies, especially CD4 binding site antibodies. But that didn't pan out. I mean, the antibody sensitivity of high CD4, low CD4 affinity viruses is indistinguishable.
Can I argue with you a little bit?
Of course.
if it's that they fuse better, and that affinity is just, a marker of better fusion, wouldn't that imply that there's something else in the CD4 T cell membrane or the macrophage membrane that somehow facilitates or inhibits that fusion? Because it's just hard for me to see how just affinity makes all the difference for infection of these two cells, both of which have CD4 on them.
Yeah. Remember that the first event that has to occur during infection is CD4 interaction. Co-receptor interaction is secondary to that. The CD4 binding is what's needed to expose the co-receptor binding epitope. So if the virus isn't able to first engage the cell, nothing else is going to happen. So that first binding event with CD4 is really a prerequisite for infection. So with a macrophage, because there's so little CD4, the virus really has to grab onto those rare molecules very effectively, and that's why for macrophage infection you see this very high affinity for CD4. Now this isn't something that we've invented. This is people like Paul Clapham and Paul Gorey and Ron Swanstrom have spent careers looking at the CD4 requirement for tropism. And so talking about co-receptor CCR5 usage versus CX, this is all secondary. The primary determinant for tropism is actually CD4 affinity. Thanks for the question.
Thank you. The last piece of work that you showed with Viviana did, showing the macrophage entry assay of all the different virus isolates after rebound, I thought that was really quite dramatic, where you could see a whole spectrum of indirectly CD4 affinity variants being induced after you stop HAART, meaning that the virus must be coming from everywhere in the body when it's coming into the plasma. It's not like one site. It must be, it really shows you the challenge of trying to cure this infection.
Yeah. If you look at some of the work from Tim Shacker, for example, I think last year, a paper from Roethlisberger. So they took individuals who stopped treatment and then did an in situ analysis of how those earliest events in rebound, how they looked. And I thought it was remarkable because if you looked in lymphoid tissue, you didn't see one little nidus of cells all of a sudden becoming activated to make viral RNA. They were occurring all over lymphoid tissue, thousands of these little spots of viral activation. So it suggests that there are many sites in the body that are just ready to make virus. The only thing holding it back and preventing the virus from spreading to other cells is the presence of antiretroviral therapy, and argues against a sort of single founder event driving viral recrudescence, that you stop therapy, a latently infected cell that just happened to be getting reactivated at that point in time now drives this explosive growth. And that's not what we see in terms of the distribution of fusogenicity, nor what he sees when you take a in situ look at the dynamics of the earliest events in viral recrudescence. Yeah.
Do we have any further questions? Or you guys just want to have coffee? Ninguém mais? Nenhuma pessoa?
Professor, uma.
Ali, pronto.
You said about CNHG. Do you think somehow we can produce this molecule and use for a possible cure for HIV?
You know, the SAMHD is integral to the DNA metabolism of the cell. So, you know, interfering with cellular processes that are a normal part of DNA metabolism is a risky venture. But I think, you know, I think there was a mention about quiescent T cells versus macrophages earlier in terms of what SAMHD might be doing. I think it's more complicated. SAMHD is probably not the only factor restricting infection of resting T cells, because if it was only SAMHD, then SIV or HIV2 should be able to infect resting cells. They have a VPX to degrade SAMHD, and that's not the case. So there's something else other than DNTP levels that are restricting infection of quiescent cells. So I think that is a target worth exploring, because there's some very powerful force in those resting cells that keeps HIV from infecting and reverse transcribing cDNA. So I think somewhere in the antiviral restrictions that counter HIV infection, there are opportunities for development of new strategies for inhibition. You know, VIF, Tetherin, SAMHD, yes. I believe that the next wave of antiretroviral therapy, if we ever get it, if we don't cure the virus first, might center on those antiviral restrictions.
So, Mario, so to make the link with Glenn's presentation, so you assume that if you have a virus that's in macrophage, it's going to, and the virus undergoes some sort of replication, it's going to trigger sting, and it's going to trigger a whole series of pro-inflammatory events. Have you been able to quantify in those monocytes or macrophages which have HIV, whether they are producing cytokines which are pro-inflammatory?
Yeah. So if you take macrophages and infect them with HIV, you don't see an interferon response that is able to prevent them from being infected. But once they become infected and the genome is established, you see an interferon response that protects neighboring cells from becoming infected. But there is nothing in the incoming particle, whether it be RNA or DNA or protein, in that first half of the viral replication cycle that triggers any of the sensing pathways. So there is absolutely, we've looked very hard that first, all events leading up to integration, there is absolutely no sensing by innate immunity. So, you know, obviously there's literature out there arguing otherwise, but we don't really agree with some of that literature.
Anybody else? It's a little bit hard from seat back people there. None? Okay.
Thank you.
Thank Professor Stevenson for the wonderful talk, and short break. Thank you.